I'm Michelle Laplaca, and I'm happy to introduce Firas Kobiassy from Morehouse School of Medicine, just down the road. And he's been there for a few years now, where he helps to run the Center for Neurotrauma, Multiomics, and Biomarkers. So I know him through my interactions with Kevin Wang and with Firas when they were at University of Florida before they moved up to sunny Atlanta. So he got his PhD at the University of Florida and his undergraduate at the American University of Beirut, where I think our provost is now, right? Isn't that, am I remembering that correctly? His research interests are in animal models of brain injury and specifically looking at biomarkers and molecular mechanisms, and also mechanisms of drug abuse and neuronal injury. He's an expert in proteomics, multiomics, and systems biology, and also in nutraceuticals, which has been a more recent interest of his. So we're looking forward to hearing your talks. Thanks.
Okay. Thank you, Michelle. It's a pleasure to be here at Georgia Tech. That's my first time here. So I'm a professor in the Department of Neurobiology at Morehouse School of Medicine. Today, I'm going to talk about MitoQuinone and as a neurotherapy in traumatic brain injury. So how many of you are familiar with traumatic brain injury? Okay, perfect. That's enough. Good. So for those who know me, my focus or my research focus is biomarkers. So I look for protein changes after brain injury, and that's has been my PhD and my interest since so long. So you will be like seeing here two biomarkers that I will discuss, and they were FDA approved in 2018 on February 14. So this is my area of interest. However, talking about MitoQuinone, and I'll dwell about this particular molecule, is because we got a grant from the VA. It's a merit grant that's assessing postsynaptic changes in brain injury and experimental brain injury after MitoQuinone treatment. So these were all preliminary data, and we have published these data. They are now descriptive, and we're going into more mechanistic. We're still working on it. So before I continue, I have a disclaimer here. I serve as a scientific consultant for Abbott, and that's because of these two biomarkers for their iStat TBI plasma test device. So Abbott has designed this small device. You can put it in the ER, and anyone with the TBI, they can check the levels of these two biomarkers, and they can either decide to go into CT scan, or it can decide not to go to CT scan. So this is very, very useful and decreasing the exposure for CT scan on patients with brain injury. And it is specific for mild TBI, which is something extremely important in our talk. So the objective of our talk today is to talk about pathogenesis, prevalence, and why TBI is important. It's traumatic brain injury. I'll go into neurotherapy and what has been done recently in the area of TBI. And unfortunately, there is no FDA-approved drug till now. Describe the emerging scientific research in our lab, and we are open for collaboration and having students as well. I'll start with the TBI history, and it's interesting. There was one professor at the Department of VA. His name is Ralph De Palma. He passed away 2 years ago, and I reached out to him with another professor, Dr. Dixon, and we asked him, would you like to write about the history of TBI and what happened? And we knew that because he is a vivid reader, and he can tell you he's very old, in the 90s. So he wrote a beautiful chapter talking about TBI history, and he was able to track it to the Iliad, which is in the Greek days. And it says, and the bronze spear point fixed in his forehead and drove inward through the bone, and a mist of darkness clouded in his eyes. So he was like describing that TBI can be traced, and it was well described since the days of the Greeks. And if we look at what Hippocrates of Kos was saying at that time, no head injury is too severe to despair of, nor too trivial to ignore, as if he was predicting the impact of what we call now mild brain injury is something that we have to care for. And that's what we are seeing, because with the NFL players, they are always exposed to something called mild TBI, repetitive mild TBI. However, some of them are developing, which is chronic traumatic encephalopathy, which is another form of mild kind of neurodegeneration to the brain. because of this mild TBI. So TBI has been described or defined as it is a disruption in the normal function of the brain, usually caused by a bump, blow, or a jolt to the head, or by a penetrating head injury. So what we take from this definition, there can be like different instigations for TBI. It can be an open head, or it can be like just a hit without having an open head. And this is like a little oversimplification of what we're looking at. TBI can impact everyone, anytime, everywhere, whether it is military, a general population, it's the veterans, everyone is exposed to TBI. So here's some epidemiology and prevalence for TBI, and we have to see that the socioeconomic burden of it is very huge. So in terms of economy, it is like approximately 400 billion is spent for TBI yearly. Approximately in the third world countries, such as in Middle East, in Africa, we have 2 million that are like just dying because not proper treatment or intervention because of TBI. And TBI represents the greatest cause of death and disability globally due to injury. And for classification, what we have on a clinical level, we classify TBI as mild, moderate, and severe. And this is, we call it the Glasgow Coma Scale. And this is a crude neurological assessment based on loss of consciousness or post-traumatic amnesia as well. So if we have loss of consciousness less than 30 minutes, we categorize TBI as mild. If we have between 30 minutes and less than 24 hours, it's a moderate TBI. And if we have like more than 24 hours and it is the severe and it is the most serious one, that's what we are looking at. But what do you think it's the most prevalent kind of, these kind of category or classification of TBI? Mild. Good. What's the percentage approximately for mild TBI? 50, 60? Okay, 65. I mean, you can go up as well. So it reaches 75? Yes. So actually, mild TBI has been considered as a signature injury and wound in the Operation Enduring Freedom and the Operation Iraqi Freedom for both the military and the veterans. And it was shown that mild TBI, which is the prevalent, it reaches approximately 80% of all TBI, is something very, very serious because it was shown that mild TBI comes as repetitive doses. So we can have like subconcussive or concussive mild TBI. However, when we have like these soldiers being exposed to mild TBI, meaning like there is a blast injury happening, or when they are driving their SUV, a blast is happening, then they have rolling of their SUV, they will look the same. Okay. However, when they are presenting on neuropsychological assessment, they will say like, I'm not feeling well, or even on their PET scan after chronic time points, they will show some kind of pathology. And it has been shown that tau protein, which is, you're familiar with the tau, it's the one protein that's prevalent in the brain. It gets either phosphorylated or hyperphosphorylated after exposure to repeated mild TBI. So this is something serious. And I was actually reading about this complication. It was considered one of the complex features of TBI. So TBI is characterized by being highly heterogeneous, and it is not a single event. So when we say like, I have a model of TBI and I'm looking at it at this time point to see what's changing, this is actually really like oversimplification of what we are looking at TBI. TBI by this one paper by Dr. Dewitt and another one with Dr. Mass, they say like, there are no two TBIs that are the same. And even when we are doing experimental models of TBI, you have the same condition for the animals and you are exposing to the same kind of model, you'll get variability because due to the complex mechanisms happening in the TBI. So imagine when we are looking at TBI in humans, we will be seeing like different factors, smoking, exposure to drugs, genetics, and all these co-founders can be like affecting the consequences or the endpoints that we are assessing TBI to be checked for. So also on the level of classification, to just make our life easier to model TBI in the animal level, we say like TBI can be open head or closed head injury, just as the definition from the CDC. So when we are saying that open head injury, we are going to say that when we have this kind of injury, it is happening at particular space in the brain. So if it is in the cortex, it's in the cerebellum, wherever you are localizing, saying like, oh, it is a focal injury. So this is open head injury. It is at one site affecting one area of the brain versus when we are saying it is closed head injury, where it is like more diffused and the cell injury occurring between closed head versus the open head is totally different. So in the case of open head injury, we see like neuronal cell death. However, what we see in the closed head injury, we see something called diffuse axonal injury, which is like one mechanistically different feature between these two. But that is, again, it is highly oversimplification of what we are looking at. I was attending a talk last week on neuropsychological changes in clinical TBI. And even when we have moderate TBI, closed head in humans, it was given by Dr. Williamson from University of Florida. So he said the site of injury, whether it is in the front or on the back or on the side, can have different consequence in patients. So here, when just we are simplifying, it's an open head or closed head without even looking at the area that's impacting, it is something like we have to look for. And on the level of animals, we have different models that can characterize open head versus closed head, and these are the ones that we are seeing. So this is in our lab, it's called the brute percussion. It is more closed head injury. I mean, open head, but it is the diffuse injury. We have the weighted drop model that Dr. Levi is working on. We are working on control cortical impact. Other labs will work on blast injury. So all of these can be like models that can characterize different areas of TBI. Again, for TBI, when you are reading textbooks or you're reading like mechanisms of TBI, it is occurring in two phases. We have the primary, secondary phase, and from this you have to see like at the primary phase, it is happening within minutes to hours. In the secondary phase, it is where you can intervene because this is so fast. And even when you are talking to neurosurgeons, when they look at intervention, it is happening in the secondary phase injury. And from here, I would like to focus on oxidative stress and mitochondrial dysfunction because these two I will be hinting to when we are looking at intervention. So oxidative stress and mitochondrial dysfunction are two areas that we focus on as neurotherapeutic targets for looking at TBI. And this is actually by Dr. Hofda. They were looking at assessing the different secondary injury pathways, and they found, or they characterized that mitochondrial dysfunction, oxidative stress, and excitotoxicity are predominant in the areas of TBI. Okay, and remember, I told you I work on brain injury and on biomarkers, and one consequence of these kind of mechanisms are the changes of proteins. So as you are seeing here across time, these proteins that involve like inflammatory markers such as CRP, autoantibodies, tau, p-tau that we were talking about, NFL, all of these are changing according to different time points in the injury trajectory. However, there are two proteins that we characterized in 2018, but the story started in 2002. They are the early time points proteins, which is UCHL1 and GFAP. These two proteins rise acutely at early time points. So if they are high at a certain threshold, you know that there will be like injury, which is serious in the brain that requires to have CT. So based on this data, actually, we were like hypothesizing that these proteins can sieve from the brain, which is the tissue, to the CSF, and from the CSF, it goes more into the blood. And these two proteins, which is UCHL1 and GFAP, are highly predominant in the neurons, which is UCHL1, and in the astrocytes, which is GFAP. So they indicate that we have cell injury occurring and requires CT positive or CT negative. So this is the implication for these two proteins. And the story, like looking at biomarkers, is not that easy. So you can see like this is here the trajectory of what we did. It started at University of Florida. It ended up with now two big companies, Abbott and bioMérieux. bioMérieux is taking the license for Europe, and Abbott is in the US. And now these machines are present in every ER to triage CT positive, CT negative on mild TBI. So utility here is only mild TBI to look at injury levels to triage for CT positive, CT negative. Okay, and this is like the announcement for these two biomarkers. And recently, we are working on areas of third-generation biomarkers, which are autoantibodies. So we are thinking that these biomarkers, antibodies, you're familiar with antibodies, correct? Everyone is doing Western blotting and doing like immunohistochemistry. So these are antibodies that are actually mounted against our own antigen in the brain. So once we have TBI, these antibodies will go into the rice. However, they are more stable than antigens, and they will stay at chronic time points for brain injury. So you can detect these autoantibodies at time points of 6 months, 1 year, and even post 1 year, which are called like remote time points in TBI. Okay, so now we will go into like real research. So TBI and neurotherapy. Currently, no FDA-approved drugs are available for TBI. 30 different clinical trials in the past 3 decades turned out unsuccessful in phase 3. So many were like proposed, let us say, like, for example, at Emory, Professor Donstein was working on progesterone, and it ended up that these are not therapeutic in cases of TBI. However, there are other reasons why these kind of drugs sometimes fail because of the heterogeneity of subjects with TBI. Okay, that's one thing. Drugs having antioxidant effects are shown to be effective in reducing negative effects of TBI. For example, N-acetylcysteine, at least in the experimental TBI. If you're interested in this topic, DOD and the VA supported a huge consortium called Operation Brain Trauma Therapy, and they were screening approximately 12 different drugs to see how efficacy they are against TBI. However, we were reading recently about MitoQuinone. So MitoQuinone is a drug that's targeting mitochondria. And we were writing like some support for the grant, which is looking at synaptic vesicles, post and pre-synaptic. And we said, let us try this for several reasons. So MitoQuinone was assessed in Alzheimer's disease, assessed in Parkinson's disease, in stroke, and ICH. And it was showing like it is highly antioxidant in nature. So we started doing like some kind of preliminary studies, starting as just any lab. Like we assessed MitoQuinone in SH5Y, which are neuroblastoma cells, which are used as models for neuronal cells. So that's what we are going to describe in the following slides. So you are familiar with the mitochondria, correct? What's the mitochondria? Powerhouse. Good. What does it have? Complexes. Are you familiar with the complexes in the mitochondria? How many complexes we have? Biochemistry. 4, and the last one is the ATPase. Okay, so we have 4 complexes in the mitochondria. The main function of the mitochondria is ATP and energy. Okay, what's the bad consequences of mitochondria? We can sometimes mitochondria under stress will secrete reactive oxygen species, okay, that are toxic to the cells or the neurons, okay, if we are talking about neural cells. So based on this, in TBI, we have high level of reactive ROS being secreted, and we see like a lot of mitochondrial dysfunction being characterized in TBI, whether it is a closed head injury or open head injury. So based on this, I mean, that's what you are mentioning. ROS are natural byproducts of the normal metabolism of the mitochondria, and it would be helpful to decrease the level of ROS. So that's why N-acetylcysteine is an optimal drug that can be used sometimes as decreasing or scavenging for ROS or RNS, reactive nitrogen species or reactive oxygen species. So this is the 4 complexes here: succinate dehydrogenase and ADH dehydrogenase. And then here, the last one, which is sometimes we call it complex 5, it's where the ATPase or ATP synthase is secreting ATP. So once this ETC, electron chain sequence, is dysfunction, we don't have ATP, and the energy for the whole cell will go down. And then what you are expecting is neuronal injury or neuronal cell death. So what's MitoQ? And why it is proposed that it is specific for the mitochondria. Okay, are you good in biochemistry? So triphenylphosphonium. Okay, do you expect to be like positive or negatively charged? Triphenylphosphonium, that's that part here. It's highly positive. And the way it is characterized or it is designed, it has 3 phenyl groups in order to localize the electrons away from the phosphate. So this phosphate, which is now highly positively cation, it is used as an attractant for anions, okay? So we have a linker, which is alkyl linker, which is 10 carbons connected to TPP. And then here we have a quinone. Quinone is a substrate for the complex 3 and complex 1. It is a substrate, meaning it is part of the mitochondrial 3 different enzymes, complex 1 and complex 2 and complex 3. But complex 1 and complex 3, they have quinone inside, okay? So when we have, let's say, quinone being administered to the mitochondria, it will be functioning as something important. I'll explain you the mode of action for TPP and the MitoQuinone. So MitoQuinone, it is in the oxidized form. When it is reduced, it's changing into quinol, okay, instead of quinone. So instead of having double bond O, which is carbonyl, it will have OH, which is an alcohol. Okay, so the change between carbonyl and OH, which is alcohol, it is reduced versus oxidized. Okay, so when this is quinone level, it is oxidized. When it goes to the mitochondria, it will be reduced into quinol. So if there is any kind of ROS production, this quinol will reduce these ROS or scavenge the ROS. So it is protecting against ROS production. Then when it is oxidized again, it will be reduced by complex 2. So that's why it is very, very important to have quinone over there, because it is shuffling between reduction, oxidation, as if it is cleaning all the presence of ROS. However, at the same time, it is activating one kind of genes, which are antioxidant genes. We'll talk about this. This is the quinone, this is the quinol, and it is going between reduction, oxidation. By doing this, it is removing all the kind of ROS in the mitochondria. So we are protecting and keeping the integrity of the mitochondria. So talking about the action here, why do we have TPP and the linker? The problem was, how do we get the MitoQuinone or the quinone into the mitochondria? So the membrane potential between the cytoplasm and mitochondria is very, very high, because it is highly negatively charged from inside. So they designed TPP to be sucked into the cytoplasm. So once it is in the cytoplasm, it is 10 times more than extracellular. However, once it is in the cytoplasm, it will be sucked into the mitochondria, which is 1,000 times compared to the cytoplasm. So now we have a gradient between extracellular, cytoplasm, and the mitochondria, and that's where we have enrichment of MitoQuinone over there, where it is now protecting against all the ROS being produced. So this is what I'm talking about. MitoQ will activate NRF2, which is a famous mechanistic signaling pathway. NRF2 will be like, it's a transcription factor. It will be activating the AREG region, which is antioxidant recognizing element, response element, which will lead to the upregulation of SOD, catalase, and the heme oxygenase, which are protective in any kind of ischemia or high ROS levels, because catalase will change H2O2 into H2O plus oxygen. So anytime we have high levels of H2O2 due to the oxidation in the mitochondria, H2O2 will go up. So one way to combat this is to make an enzyme that can change the H2O2, which is hydrogen peroxide, into H2O plus oxygen. And one way of doing it is by activating the ARE, and ARE will activate or would lead to the transcription of catalase and SOD that will lead to the reduction of H2O2. So this is what we plan to do, like to try MitoQuinone on cell culture neuroblastoma, which is SH5Y, in order to see like production, so neuroprotection. So we had different aims here, and we were assessing survival, oxidative stress, and the level of catalase SOD levels. And at the same time, we were looking at cell cycle that will be changing after a treatment with MitoQ and H2O2. So for the first set of experiments, we did what we call dose response, and we looked at the optimal concentration that would be protective for cells exposed to H2O2, pre and post treatment for H2O2. So these were like proof of principle. So when you are doing any kind of experiment, before going or investing in animal model, you have to see like, is it protective or not? Okay, so that's what we were conducting in the lab to see if neuronal cells would be something that would be benefiting from using MitoQ, and at what levels we can use MitoQ to be protective. So this is what we did. Like we use a model for neuronal cells called SH5Y, and then we exposed these and we looked at cell viability, morphology, and we looked at membrane potential for mitochondria, fission fusion of mitochondria, and also we looked at cytoprotective genes, upregulation or downregulation. So this slide is very, very important for you who are working in areas of cell culture, looking at dose response changes with the drug and with H2O2, for example, as a toxic compound. And at that time, 0.1 millimolar or 100 micromolar was shown to be like optimal for this kind of treatment. Okay, and here you can see the differences when we are treating with MitoQ with H2O2. You see how healthy the cells are versus how toxic it is when we have H2O2 alone. So at this level, only like looking at LDH and looking at different endpoints such as looking at MTT. So who is working with the cell culture here? Okay, around 3 of you. Okay, so these are like different endpoints looking at survival. And then here we looked at cell viability and growth inhibition for these. So we did like injury model of the cells, and we looked at MitoQ. It is proving to be pro-survival, and it's working. And here, actually, in this slide, we were looking at cell cycle, and we found out that H2O2 treatment will keep the cells in the S0 phase, versus when you are treating with MitoQ, it will lead like to regeneration and proliferation of cells similar to the, or approximately, it has similar profile as the wild type without treatment. So it is surviving. It's not the same as the control, but there was some kind of proliferation occurring in these cells treated with this drug. And then what we did more is looking at fission and fusion and membrane potential for the mitochondria. And we did seahorse assessment. Are you familiar with a seahorse machine? So this machine is actually, it will look at the oxidative stress, oxidative phosphorylation in the mitochondria, and it will look at the levels of ATPs. So it is similar to doing an ELISA, but it will look at all of these indices in the mitochondria. So we found out that actually MitoQ, with all these data, are supportive to be as claimed. And these are more like looking at the mitochondrial network. You can see clearly here how it is in the control versus H2O2. We have disappearance of the mitochondrial density, and it is now more blebbing compared to when we are adding H2O2 with MitoQ. It is at least keeping the integrity of the mitochondria clean. And then looking at NRF2 genes and catalase and superoxide dismutase, we found out that they are actually upregulated after treatment with H2O2. So that's how mechanistically it is proving to be protecting these cells after like exposure. Okay, so with this, we ended up with this conclusion that cell viability is enhanced, mitochondrial protection is provided, and we have oxidative stress to be decreased, as well as inflammatory response to be like ameliorated upon exposure to H2O2. So this actually brought us to the second question: can we go into animal models of TBI? And if you still remember, we have two kinds of TBI models. What are they? Open head, and the other one is closed head injury. Open head injury is mimicking what, if you think in real life? Yeah, but if you have an open head injury, that means like someone who is more hit by a bullet, for example, or he had an accident and he has a fracture. In case of closed head injury, it is similar to an NFL player or sports exposure or athletes. Okay, so here what we were thinking is MitoQ administration in open head injury, and we used a model called closed head controlled cortical impact to model this. We will see this model here, and we will look at cognitive and different behavioral tests to assess the neuroprotection. We looked at cellular response in terms of neuroinflammation, and we looked at the enzymes that are changing. At the same time, we are conducting mitochondrial changes. I'm not presenting it here, but I'll go and tell you about our experimental design. So here, what we did is we conducted the control cortical impact. It is a model for open head injury. You'll see it clearly. I have a short movie. And after that, after 30 minutes, we started giving the drug for 30 days, and it was 3 times per week. So that means these mice will be receiving 3 times MitoQ every week. But the first dose that was given, it is after 30 minutes after initiation of the TBI, traumatic brain injury. Okay, so here is the machine that we have. It's called the control cortical impact. It is highly reproducible. We like it a lot. It has different variables. So we have something called the dwell time. So that means it is an impactor that goes into the brain. Dwell time is how long it stays in the brain. For example, 0.1 second. So it is staying inside for 0.1 second. Velocity, how quick it goes into the brain. Depth, how deep it is. And the depth here will be determining to me it is a severe, it is mild, or it is moderate. So the deeper I am inside, it means it is more severe compared to just being on the surface. So what we did here, it is moderate to severe TBI. It was 1.5 millimeters. And if you look here at this short movie, this is the impactor at the side. And if you look at it, so here you have a skull taken, and it is like you see that the impactor will go there and it will stay for 1 second. Actually, I think it's much less. And this is how it looks as a cartoon. So basically, when we have the CCI, control cortical impact, it will make a groove in the brain. And we selected the area which is parietal cortex, because we want to study some kind of behavioral tests that are relevant to this area. So we did the neurological test, and these include like a battery of tests, which are 12 different indices. We'll discuss them later. We looked at the other behavioral tests, pole climbing, to look at animals or mice, how they have balance. We had the grip test. From the name, you have to expect that we are measuring the muscle strength. Adhesive removal test, it is how attentive are these mice. And then we looked at novel object recognition to look at the learning and memory. We did the force swim test to look at depression. And we looked at memory, which is through Morris water maze. Any question about these? They may sound like a little complicated, but you will see them in the coming slides. So these are how we are conducting the Garcia neurological test. So it is a battery of tests. These are like you're looking at the animals, how fast it will flip to the other side after injury. When you are pulling his whiskers, how responsive it is. When you pull his toe, it's like moving or not. So you get the rubric. The less number, the more severe it is. Okay, so this is how we are conducting the Garcia neurological test. This is the grip test here, and we are evaluate the muscle strength, both like to test the muscles of the hand. So this is measuring the gram force of this animal. This is the pole climbing. It measures motor function, motor coordination, balance. So basically, you put the animals on the top, and you see like how fast it can rotate, and you measure it, how long it will stay there before coming down. Some of them with TBI, they will fall. After moving here, they will not have balance to go down. And then you can also calculate how slow they go into the middle, which is marked here. So this is the pole climbing, one of the easiest tests, but it gets you like good endpoints to measure. So you can see here, that's the first set of data that after 3 days, 7 days, and 30 days, that the ones that animals taking MitoQ, we see like good enhancement compared to the one with TBI. And here, when you are looking at different marks, such as adhesive removal, like you put a Scotch tape on the whiskers and you measure or the time taken by the mouse to remove that by its forearm. Okay, so it shows that the CCI is taking the longer time compared to CCI plus MitoQ which is therapeutic here. And then looking at the grip strength, we see like gaining of muscle strength compared to the CCI, which is the injury one. Also, what we did here, novel object recognition, which is measuring anxiety. So you put the mouse in a box and you measure the localization. If the mouse is actually have the ability to rotate around and investigate areas that's indicating that anxiety is less compared to the ones that are impacted. This is measuring here, that's the heat map. And this is here actually looking at the Morris water maze. So you put the animal swimming in around the swimming pool, and it is like measuring to know the place of the rod, which is like it will be trained for it. So this is something like we conducted on these animals. And finally, we did like the force swim test. You measure how long for the animal to be able to swim compared or going to drown versus the one between CCI and MitoQ. And then we moved into gene expression, looking at the expression level of markers of antioxidant markers, as well as markers of the mitochondria. So we measured these either on the protein level or doing RT-PCR, and we found out that NRF2, which is the pathway that's proposed for MitoQ, is upregulated in the cortex of these animals, in the ones that are taking the drug versus the ones that are either TBI or the naive. So this is something interesting for us. Next, we looked at the neural expression for certain cells in the brain. So what are the major cells in the brain that we look at usually? You are in the neuroscience here. What are the most famous cells that you can think of in the brain that we can assess? Astrocytes, neurons, and what else? Glial cells. Well, glial, they are the microglia. Okay, so these are the three cells: the microglia, astrocytes, and the neurons that we will see, like if they are changing or impacted after having like this kind of injury. And also, what we looked at, these are actually the cells in the brain, and we looked at certain markers inside these cells. Okay, and this is actually a very nice slide. It shows that in the cortex and hippocampus, we are seeing like changes in the microglial cells. And you can see like how inflammatory cells are upregulated in the CCI, and this is actually normal. That's not novel. But what we were happy about is that looking at the microglial cells in the MitoQ cell, in the MitoQ treated cells, they were less in number. Okay, same applies. Yes.
The question about when you apply the MitoQ, is it one time after the injury, or is it kind of continuously until the time that you do?
So the first dose of MitoQ was given 30 minutes after the CCI. However, over 30 days of treatment, we were giving it 3 times per week at 8 milligrams per kilogram.
This is 30 days.
Yes, this is 30 days. Yeah. This is the terminal time points. Same applies for looking at GFAB astrocytic markers. So this GFAB is a marker for astrocyte upregulations, or at least recruitment to the site of injury. And you see like definitely less with the MitoQ treated versus the ones that are TBI. You can see in the last row down, you see TBI versus MitoQ versus the control. I mean, always the control or the sham animals, they have less astrocytic markers. Here, when we are looking at neuronal cells, we didn't see much difference, statistically difference, but we found like, at least in the case of in the hippocampus and in the cortex, we saw that NeuN actually is higher compared to the CCI animals. Okay, here actually we are looking at myelin basic protein. Myelin basic protein, it is a marker for the dendrites, and we see that we see shearing in these dendritic markers. If you compare between the control CCI and MitoQ, you can see beading and non-continual MBP versus the ones that are injured versus the one that are being treated. So this is encouraging, and we propose that MitoQ will be working on neural cells, at least decreasing inflammation, okay, and enhancing the NeuN stability and integrity. In addition, it is controlling the level of certain cytokines, certain antioxidant markers. The next set of experiments, or the question that we were asking, is relevant to the grant that we are doing, closed head injury. And again, closed head injury is 80% of all TBI. Closed head injury, repeated closed head injury can lead to what we call CTE in some instances. For you have seen the movie Concussion. We have seen it. Okay, Michelle is the only one. Okay, so basically we asked the same question that we did for open head injury to be like asked in the closed head injury, and here is what we proposed. So we did 3 times injury that resembles or mimics 3 hits at one site. That's different between like one hit, another hit by 24 hours. Okay, and that's actually a slide that's very, very important, and I think we can use it in the grant. So this is, we were measuring the tau and phospho-tau, comparing 1 hit versus 3 hits at different time points. So here we have day 1, day 3, day 7, and day 10. And we found out that across time points, we found that tau and p-tau, p-tau meaning phosphorylated tau, it goes up much higher levels in the case of repeated closed head injury. So you get the animal, you expose it to 3 times closed head injury, so that means we are not fracturing the skull. We are hitting the skull from outside, and that will lead only like, or mimicking sports injury, or what we are seeing in the army or in the veterans, they are exposed to repeated concussive or subconcussive kind of injury. So these 2 markers are very sensitive to indicate that really we have changes in these biomarkers occurring. So this is here actually the modified closed head injury. What's different between the CCI and this model is we don't have fracture, or we didn't like open the skull. Okay, so this is how it looks like. I don't know why it is like this. So we hit the brain 3 times. So this is the difference between 1 hit and another hit is 24 hours. However, you can modify this kind of repeated hit according to the model that you are thinking it is relevant to you, the question you are asking. So you can do this kind of injury after, let's say, 1 hour, 1 hour, 1 hour, okay? But you have to be very, very careful in conducting this closed head injury, because if it goes like less posteriorly, then it will be hitting the brainstem, and you can expect that this animal can die. So it's not as easy as you are seeing here, but it is a good thing that you are seeing. So one thing that we found here, which is different than the open head injury, the motor function are less susceptible compared to the open head injury. That's why we use this test, which is called rung test, which is ladder rung test, to look at the fine motor coordination in these animals. This was not actually impacted in the motor function compared to the open head injury. So here it was like we use this test to show like we have differences in the fine movement of these animals. So any like missing of these steps was recorded, and we got like a rubric to see how these animals are compared after MitoQ treatment versus closed head injury, which is 3 times. And again, here we were like repeating similar paradigm as we saw in the other one. So the different score we got like a total missing, deep slip, slight slip, replacement, correction, and partial missing. Yes.
So did the closed head injury also have deficits in the neuro score?
Yes. Yeah. I mean, what I got here, oh yeah. So these are the changes that we are seeing, but it is different than what we saw in the open head injury. It's less predominant. We didn't see like much differences, except in the ladder rung test, which is very, very sensitive compared to, let's say, you're doing rotarod. Are you familiar with the rotarod? So what's rotarod? You say, it seems like you are doing rotarod in your lab, the one behind you.
Me?
Yes?
That's the one where the mice have to hold on and you rotate the rod and see if they can actually stay on it, right?
Exactly. So we are measuring like the running of the mouse or the animal, maybe it's also rat. So here it shows like closed head injury, even with the repeated ones, 24 hours, we didn't see like pronounced deficits in the neurological scores, except with the ladder rung test. MitoQ rescues and RF2, we saw like similar kind of phenotype as we saw in the open head injury. We did the extra staining, which is called DHE staining. It's very easy to conduct. It looks for ROS, and it stain as red. You can do quantification. We did MAP2 which is another marker for dendrites. We looked at GFAB, IBA1, and all of these were changing similar to what we expected, however, to the less level compared to what we saw in the open head injury. We saw like myelin basic proteins also being beaded after repeated injury. That's at chronic time points. And what we did here, we looked at acute, which is 1 day. We looked at subacute, we looked at 7 days, and also we looked at 30 days. So here we have a continuum of injury follow-up versus the open head injury, which was 30 days. Also, we looked at new N survival, and we found out also like new N is not highly impacted in closed head injury. So this is a remarkable distinction between open head versus closed head. So we didn't see much death of neurons in the closed head as we saw in the open head injury CCI. So this was comparable or comparing the early time points, acute, subacute versus the chronic, which is 30 days. So here, I know you are feeling like little bored, but here you'll get more exciting stuff. So the last, yes.
Dave Dean, who asked, are the physiological outcomes that you've seen, excuse me, seen here expected to occur generally, like not specific to TBI?
Well, these were targeting TBI, but we see them specifically with TBI in terms of behavioral deficits and neuroinflammatory response. But yeah, I mean, do you think, what is he referring to?
It just says generally. I asked him.
I mean, we can expect to find similar deficits in stroke or Alzheimer, for example. I know several studies have tried the MitoQ in Alzheimer's studies, and one of them is like recent, and it showed like these behavioral deficits are also pronounced. So you expect to see something similar. I mean, these animal models will test the same thing in general. Okay, other questions? Okay. So part 3 of the study is to look at mechanistic changes that are relevant to proteins, and that's where we looked at omics changes, and we looked at glycoproteomics because our collaborator was asking, can I do glycomics or glycoprotein? These are proteins that are changing with the levels of sugar moieties on them. So here the hypothesis is the protein is not changing. However, the sugar level on these proteins are changing due to a certain modification. We all know, like after transcription, we have translation. The protein as is can be subjected to different kind of post-translation modification. What's the most famous post-translation modification? Many of you are studying here in as part of your lab research. The MAP kinases, what do they do? Kinases, what do they do? Phosphorylate. Where do phosphorylation occur? 3 amino acids, famous ones. One of them is tyrosine, serine. Very good. Threonine, tyrosine, and threonine, and serine are the 3 amino acids where phosphorylation is occurring. As for cases of glycomics or glycoprotein changes, they are more and more complicated because they look like Christmas tree. You'll see like all kinds of sugars added to these proteins, and it's so famous in cases of cancer. So these proteins, when they are modified, they become receptors for other homing cells. So macrophages or metastatic cells, when they see this kind of modification, they will use these sugars to go inside other organs. For example, metastatic from the liver to other organs happens through these kind of modifications. So we were thinking, do you think that this modification can serve as markers in cases of different severities of TBI on the clinical level and also experimental level? So that's what we did. We ended up with excellent data that discern the changes of treatment versus non-treatment, like TBI, MitoQ, and control at 3 days, 7 days, and 30 days. That's enclosed head injury. And these are the different proteins, I mean, and we did a lot of validation on these proteins. At least they can hint on the mechanistic role of MitoQ in these animals. And that shows, like, for example, in cases of 3 hits after injury in the cortex, we see a lot of pathological pathways being altered. These data are from our animals. We see like cell migration, dendritic cell migration, complement activation, and these are relevant to what we expect because neuroinflammation is a major thing we see in cases of TBI. However, after MitoQ, we see a lot of therapeutic pathways or biological processes that are altered, including neurite outgrowth, neural plasticity, as well as neurodegenerative diseases that are relevant to these proteins, such as we saw here tau, we saw UCHL1, and we saw GFAP. Okay, so we ended up publishing this work, and this was one of the schematics that we presented in this work. We have so far a number of publications on this area, and one of them is in Frontiers for Young Minds, and it's simplifying the concept of mitochondrial injury in cases of TBI. Current projects in our lab, this is one of really, like, I think this is, I'll give it 1 minute. So this project, actually, it is as if doing an atlas assessment of all the proteins that's happening after closed head injury. So we are doing this with the University of Lille. So they have a center for omics, okay, and they accepted to do like this kind of characterization. It required a lot of machine learning and changes of proteins occurring at 48 hours, 7 days, 30 days. Okay, and then after that we have sham, single hit, and repetitive hit, and at the same time the third dimension is looking at different brain regions. So putting all of these dimensions together actually will give you like a nice overview of the whole proteins, how they are changing, and why they are changing, and what do they represent. So this is actually already in process of second review in one of the journals. This is a very hot data that we received last week. So we are doing something called spatial transcriptomics, and we are comparing fluid percussion injury, which is, as I said, it is one of the, you open the skull, but it is a diffuse injury. So it is not hitting at one side. It is pressuring saline in the brain, so it's hitting the brain as a whole. So we call it diffuse injury. We are comparing the gene clusters that's happening at different areas of the brain with the different clusters of the genes. So this is highly like high throughput data and very complicated and complex data that tells you how genes are changed. So if you compare the fluid percussion versus the control, you can see the heat map, the 3D heat map changing among these two. So this is, we're waiting the different clusters from this work. Also on biomarker side, we are working on something called lateral flow. And this is we designed during the COVID because the clinical trials in TBI are cumbersome, or even any kind of biomarker studies. So collecting 10 ml blood from a patient and then taking it serially after he's being like discharged from the hospital is something very challenging. So at that time we thought, like, can we develop something called lateral flow that will capture serum or blood, and then it will be like stable, and it will be shipped to us to do biomarkers. So in our lab, we do a lot of biomarker assessment with state-of-the-art machines that can look at these two biomarkers, GFAP, UCHL1, and others, and tau. So this was shown to be optimal, and this is how we actually, package it. So now we have a clinical trial happening with the Emory Neurosurgery Department, and this is funded by the DoD. So we collect blood. We are doing assessment between wet plasma, which is the patient real samples, versus dry ones, which is lateral flow. So this is another project happening, and this is what we are presenting the data at the neurotrauma and also at the military conference in Florida. And here we did this kind of collaboration with what's her name? Raquel Gardner. So she sent us blinded samples, and we separated them between wet and dry, and we compared the results. It was excellent kind of correlation between these two. We did the omics on them to see the profile. If it is compatible with omics study, it turned out to be successful. So we are working more on this work. Final project we are working with is also biomarkers of PTE, which is post-traumatic epilepsy, which is a feature of brain injury. Some of these animals, I mean, in our case, we are using one CD1 mice, and they develop 50% of them, they will develop post-traumatic epilepsy. So we are thinking why this cohort can develop epilepsy. So this is what we are looking at. And this is from multi-omics point of view. So we are looking at metabolites, protein, transcriptome, and also microRNA. So this is like the different cohorts that we have. It is like 3 controls versus 2 injuries. And finally, like looking at PTE, which is PTE autoantibodies. And these are antibodies that can be secreted by our immune cells against our own antigen. So here, in cases of brain injury, there is this regulation of the immune response that can see some of our antigens as foreign antigens, and that can exacerbate the whole consequence of brain injury, or what we see in cases of post-traumatic epilepsy. With this, I would like to thank you all and thank Michelle for hosting me. And this is definitely our lab in Lebanon. Most of the work was done over there. And many of these are now doctors, actually, and some of them are here. And I would like to acknowledge several collaborators from University of Lille, Dr. Michel Salzat, Texas Tech, we have Dr. Mishrif. At Mario Negri, Elisa Zanier, she's helping us on similar drug, doing MRI on them. At Walter Reed, we had Dr. Debshir and Jignesh. They are very highly into mitochondrial therapeutics. So they are testing at 2 different sites the same compound that can reflect our rigor. And they have actually nice data over MitoQ and another drug. At American University of Beirut, we have Dr. Mantesh and Dr. Haidar and Anefi and Dr. Jihan Swade and Dr. Khouri. And at Morehouse, you can visit us and see our team. We are welcoming you all over there. And here are some resources if you're interested in TBI. Several books are published with our teams and prominent figures in the TBI field. And I'll take any question from you. Thank you.
At the beginning, you showed, that beautiful plot that shows, how certain protein levels go up, kind of acute phase versus subacute, and then maybe decline.
Yeah.
And that's part of your motivation for targeting mitochondria, as I understand.
Yes.
I was curious whether or not you think that application of MitoQ during that entire 30 days is necessary for the therapeutic effects you see, or whether you think targeting that, kind of acute to subacute phase would be sufficient for kind of.
So let me tell you a story about MitoQ. MitoQ is one of these supplements sold at Walmart and CVS, and it's one of the most expensive one. So it is under the name CoQ because it has the 10 carbons on it. So I think taking it on longevity, that's where you get the most benefit. And they think it is actually decreasing oxidative stress and helping decreasing or occurrence of Alzheimer. So as you are thinking getting it only at acute time points, I don't think it will be highly therapeutic only. You need to take it over long range of treatment. Because even thinking about it, when it was developed by Dr. Murphy, it was a PNAS paper. It goes into the heart, the liver, the muscles, and to the brain. And in order to make it more penetrating the brain through the blood-brain barrier, they added this long TPP plus the 10 carbons. So the amount that you take and reaching the brain is minimal compared to other organs. Remember, it goes through the gut and goes then to the liver. So definitely the biggest portion is going to be to the liver. And whatever left over is going to reach the brain. So from this perspective, I think that's why giving it over long time points is the only way that you can see kind of changes with these kind of drugs. I mean, even clinically, I was reading clinical trials. With clinical trials, there was no therapy seen. They tried it. Actually, there is one happening now with MCI, mild cognitive MCI. They didn't see toxicity. Yeah, I mean, that's actually, it's demonstrated. and they are ongoing. It's still ongoing. So they think that it needs longer time to see efficacy compared like, oh, stopping after 6 months or 1 year. So still they are not seeing huge difference.
Even if you, someone is taking this over a long period of time, is there a point at which you think it's too late? You know, it's too late just because of, you know, those spikes that you see in the proteins very early on.
I mean, if you're thinking about TBI, let's say
Yeah, sure, like, you know.
I mean, it would be
later.
therapeutic. I mean, the mitochondrial oxidation is well-known feature in TBI. So I think taking it may be helpful. But again, they tried vitamin D before, vitamin E, and there are lots of these kind of intervention. Whether it's therapeutic, I don't know, like, and again, in human, it's so variable. If someone is smoker, as if he is like removing all the effect of these MitoQ and acetylcysteine or apocynin, these are all in the same category, antioxidants. But at least in animals, we're seeing some hope.
So how do these doses compare to like the stuff that you get over the counter?
Oh, I mean, over the counter, I'll tell you, you see the tablet is so huge compared like to the others. It's so huge. I think because if it needs to go to the brain, I think they put a lot. The amounts is variable. And even if you want to do like a study, there is like, Murphy was the one who discovered this or synthesized it. I think the company now divided. It is one in Australia. The other one is, I think, in Europe somewhere. And if you approach them, they will give you like different doses of MitoQ saying that it will be relevant to humans. And they will give you placebo ones if you want a clinical trial. I mean, they want to promote it more and more. But the only thing, they didn't see efficacy with these clinical trials, again, with TBI stuff, same. Because I think all these kind of heterogeneous, I mean, if someone is like living in low, in drugs, smoking, all of these can affect, or even lifestyle.
I was thinking more like if you were taking it, like you take turmeric or something like on a daily basis as some kind of supplement level.
Yeah,
Would the idea be like when someone sustained a TBI, whether they were taking it or not, you would give them high doses of it? Or would there be toxicity associated with high
oh, they don't. They
doses?
actually they didn't see any toxicity with high doses, so that's promising. That's one thing
Would
they
they
can
have
teach.
to have been, like, are people promoting it like they do fish oils where you're on it kind of in a preventative way? Or just in a generalized
The way it is,
antioxidant?
okay, that's a good point. So they are promoting it as anti-Alzheimer supplements.
Preventative.
Yes, so you take it, oh, you have less oxidation, less injury. I mean, to the public, oh, your cells will stay intact compared to someone who is not taking, and similar to fish oil. Oh, you're taking it and it's good for the brain. Same thing here. So this, I feel it's good because, I mean, the price is very, very high compared to other kind of supplements. No. Yeah, but we have it in the lab if you want some. I mean, to try it, not to, I mean, to animals or anything. Yeah. The questions? Good.
Thank you.
You're welcome.